Panama has more clinical experience with MFCs. We have these miracle cases, and then we have other cases that aren't so miraculous. Let's find out what the difference is. We started screening every donor for the presence or the absence of these different markers. In infusions, we never had even one serious adverse event.
Please welcome Dr. Neil Riordan on stage, my hero.
They're making me cry like that. I love coming to these things because I get all kinds of information that I don't necessarily get. I love going to Panama because then I get to hear people's stories, and that just lifts me up from whatever's happening in my life. And I just an example. Russell came up to me. He said he's 71 years old, and he had 17 back surgeries. He was in terrible pain. And he came to Panama a month ago, and he said, "I'm just here to tell you, the first time I had pain in 20 years." So that's pretty cool. So makes my day talking to all these people that have been down there.
That's an amazing story. Yeah. Thanks for sharing.
Yeah.
So I promised this was going to be an intimate conversation with you. So I will start asking you a personal story.
No, it's good.
So you had a big health scare in 2020,
Yeah.
and stem cells played a huge role in your recovery.
Yeah.
Can you just walk us through this experience?
Yeah. Well, I'd like to start by saying I think everybody should get their vitamin D level checked because with COVID, if you have the vast majority of people that were hospitalized with COVID had a vitamin D level of under 30, and almost everyone who died from COVID had a vitamin D level below 20. And I had shortness of breath and a little bit of a fever and in March of 2020, and talked to a doctor friend of mine, and actually down here in Miami, and he said, "Well, you better might go get checked out." You know, he goes, "How's your sense of smell?" I'm like, "I can't smell anything." So I went to the emergency room, and then the guy, and he said, "Don't take any anti-inflammatories, don't take any steroids, just write it out, and then when you can't breathe anymore, then come back to the hospital." And that came true, and I couldn't breathe. My son took me to the hospital. I don't recall any part of it. I don't recall getting there. I recall once I was there, calling a friend of mine who's a cardiothoracic surgeon, and I sent him a picture of me on 6 liters of oxygen, and my blood saturation was below 80. And I know, as a physician assistant, this is not compatible with life, and we're going in the wrong direction. So long story short, I wake up 3 weeks later in a different hospital, and they had to intubate me, and then they had to take my blood out, and a procedure called ECMO, extracorporeal membrane oxygenation. And unbeknownst to me, they had treated 37 people with ECMO, same condition as me, and they had all died. And so I was the first ECMO patient to wake up for a COVID ECMO patient at Baylor, Dallas. So I consider myself to be very, very lucky, and I usually get emotional when I talk about this, so I'm not going to talk about it anymore. There is a point to this story, and that is I went to my infectious disease doctor. His name is Dr. Spock. I'm not kidding. So I wake up, this is the funny part. I wake up, I'm psychotic. Apparently, I had been waking up for 9 days before I realized that I was even there, which is a relatively common thing for somebody who's been in a coma, is that you're there and you're talking, and then you go to sleep, and then you wake up, and it's like Groundhog Day. I had Groundhog Day for 9 days. And it wasn't until they let my daughter in at the insistence of the Governor Abbott in Texas. She's also a healthcare professional, and they weren't letting anybody in the hospital. This would have been April when they let her in. And it wasn't until she was there that I had self-awareness. But so long, hard rehab. I got some experimental treatment in the hospital. That's probably why I survived. But get your vitamin D level checked. I don't think we're having near the problems that we were having, but it's such a, it's to me, it's criminal that our medical profession do not tell people about this correlation. Just because it's correlation doesn't mean it's causation, but if you had a vitamin D level over 30, you're probably not going to the hospital. If you have it over 20, you're probably not going to die. So I was right on the borderline. But on follow-up, I went to my Dr. Spock, and you know, he does the assessment, and he said, well, you know, it's a miracle you're alive. And he goes, you know, you're not supposed to be here. So he said, what are you going to do? And just at that moment, I said, you know what, for Dr. Kaplan, for all the people that have been fighting this fight for all these many years, I want to get a product approved in the U.S. I want umbilical cord MSCs to be available in the U.S. So I hit 65 in October, and I'm not retiring. So still going.
Luckily for us.
But anyway, I didn't answer the whole COVID thing, but I will tell you that I've done multiple rounds of umbilical cord MSCs in Panama, golden cells, and I feel I'm completely. I don't know if your brain is ever as snappy as it was 5 years ago, but I feel like I'm pretty normal. Physically, I'm great. My lung capacity is back the same. I have no adverse. My renal function is fine. Kidney function, liver function, heart function, everything's great.
So talking about that, let's take you back into the beginning of your story in Panama. When did you start your clinic in Panama? How was that process for you?
Well, actually, Jorge, Dr. Paz, he said we'd known each other for 14 years. We've actually known each other for 17 years. It's just so much fun. The time is zooming, right? So I went there in 2006 and I met Rodolfo and Georgie in 2006. We actually went with a patient that we treated in Costa Rica who was a medical doctor from San Antonio, and Georgie practiced in San Antonio for 11, 12 years or something, right? And so his kids are actually American, and then they basically saw the growth and the opportunity in Panama, and Lisi's here, his wife, and they packed it up and went back to Panama about that time. He had been there about a year when I got there, just something like that. And so when I went down there to talk to them, like a lot of people say, oh, we want to do this, and I've heard that so many times, the different countries and doctors and stuff. These guys, 6 weeks later, they're on a plane back to see me, and they said, oh yeah, we went to the government, we got our lawyers, and we found this bill, this law that had been passed in 2004, and it's essentially a page. The first paragraph bans embryonic stem cell research and treatment with embryonic cells, but adult stem cells are okay to be researched, and they can be administered by a doctor in Panama. And I was like, I read it 14 times, and I couldn't believe it. We couldn't believe it, right? So that's when, that was in 2006, and then in 2007, we applied to get into a special economic zone, which is called the City of Knowledge. And we were admitted in the City of Knowledge. It gives us several benefits, not the least of which is the ability to bring in talent from other countries. And that's like hugely important because you're going to a country with maybe not a lot of experience. I think now Panama has more clinical experience with MSCs and manufacturing facilities than manufacturing, like the number of life years of people's experience is enormous compared to most other countries. And so based on all those reasons, we got invited to the City of Knowledge. The president at the time basically wanted us there, and he told Rodolfo, he said, get that guy down here, let's go. So we kind of had the red carpet rolled out for us, and we took advantage of it, and we started there in 2007, very modestly. There were just the 3 guys, and we had 1 employee in the lab. That was it to start with, and then we got a secretary, and then so we went from there to over 100 employees. Last year we treated 2,000 people, and every year, with the exception of COVID, every year we've just grown and grown and grown. Thanks to a very dedicated team who is super diligent, and knock on wood, we haven't hurt anybody, and we've been doing it for a long time. And anyway, that's how we got started.
Excellent. Everyone here received a copy of Dr. Neal's book, right? Yeah. So in your book, you talk about golden cells. What are golden cells, and what is your process for selecting cells for treatment?
Okay, well, so the golden cell thing came about in 2010. We actually had a patient from Dallas he's a 3-time state champion high school football coach in Texas. That means he's sort of way up there, and particularly for the people in the community. And he had crippling MS. He was in a wheelchair. He had been essentially decommissioned, and he was on disability. And the community basically rallied around him and said, "You need to go to Panama and try these cells." He came down, and his name is Coach Sam Harrell, and there's probably a lot of stuff on the internet. His son was actually an NFL player. His son, I think, is a coach now. But like NFL, is he an NFL coach? Yeah. So anyway, Sam comes down, and he gets this incredible response, right out of the chute, out of his wheelchair in a month, and exercising and all this kind of stuff. And that lasted for about 14 months, and then he started coming back down again. So then he came back for treatment, and he got a little bump, and historically, his history has been hijacked a couple times on this, but we do have data that he did improve somewhat after the second treatment, but it wasn't near the benefit that he got from the first treatment. Then the third treatment in 2012, boom, it was like the hand of God touched him, and there are videos of him run up downstairs, and basically he went from being completely disabled to now he's back coaching at the same place, and here we are, 14 years later, he's back coaching at Ennis High School, where he was, state champ for all the time. So I'm curious, and I was like, well, what's the difference between 10, 11, and 12? Pretty obvious question, isn't it? So, and Dr. Paz mentioned that we have samples of we always hold back samples, and we're running into a storage problem now because we have so many samples from so many treatments, but we keep some from everyone. So I called them golden cells for lack of a better term. I said, look, guys, we have these miracle cases. Then we have other cases that aren't so miraculous. Let's find out what the difference is. So I went to all the doctors, and we sat down, and we said, all right, give us your top miracle cases. This was in 2012. So we got a list of 20 patients that were just like surprisingly, crazily benefited from the treatment. And so we selected, and of those, there was commonality in one of a couple of the cell lots. So we took those cell lots. We took 6 cell lots, meaning a cell lot can treat hundreds of people, and new technology has allowed us we can treat millions of people from a single lot. But back then, we could treat hundreds of people, and so we tested. I'm trying to not go geeky scientist on you here, but we basically took those cells and we grew them up, and we grew them in 2 dimensions, and we grew them in 3 dimensions, because we know that ultimately you're going to have to grow in 3 dimensions. So we grew them up, and that whole process took about a year until we had all of the cells grown in 2 dimensions, 3 dimensions. We took those golden cells, or those cells that had golden effects, and then we took cells that didn't have such great effects or mediocre effects, and then we took fibroblasts, which they're healing cells, but they don't secrete a lot of the stuff that the MSCs do. And then we ran an analysis of what molecules were in there, and it's amazing. If any, I know who's a fan of statistics in here. Nobody. No, well, a couple of you. I got you. No, I just kidding. So, a p-value, which is the probability of something being chance. Typically in science, we look at a minimum of p05, so that'd be the difference between this data set and this data set, and that's a 95% chance that it wasn't chance. And if it's a p01, then there's a 99% chance it wasn't chance. And if it's a p001, it's a 99.9% chance. So the more zeros before the 1, we had, of some of these molecules, they were 14 zeros before there was a single 1. And this was the difference between golden cells and I guess, middle of the road cells. So starting that, right then, we started screening every donor for the presence or the absence of these different markers, and then we were able to screen. And so from 2000, really, I think it was probably 2013 when we got it all finished, and we developed the tests. Then from then on, we've only used golden cells. So before it was crapshoot because we didn't know what we knew, and then once we learned that, we instituted we're only going to use golden cells. So we throw a lot of cords away, which is expensive because you wind up, you already have the expense of isolating them and everything. You have to do a certain amount of work before you can test them, and so that's what I call golden cells.
I love that name, golden cells.
Well, I wanted to name my book. So we're going to talk about magic juice here in a minute. I wanted to name it Golden Cells and Magic Juice, but they wouldn't let me. So the magic juice, that's our Kansas vernacular, I guess, for the secretions of the cells. So we call it magic juice.
A recurring question we've received was, what conditions do MSCs work and which ones they don't?
Okay. All right, well, that's a big question. Well, I think anything that is, the vast majority of autoimmune diseases respond quite well. There are different levels. So if I were to pick one for being the easiest to treat, I would say Hashimoto's thyroiditis, which affects 14 million people. 80% of them are females. And I don't know of a failure yet with Hashimoto's. And so it's so gratifying when everybody gets better, right? So I'd say Hashimoto's would be number one as far as responsiveness. Then rheumatoid arthritis would be number two. And MS, we probably treated more MS patients than the other ones. And it really depends on where they start. So I'll use the example of Holly Huber. She's been getting treatment since 2008. And I actually wanted to research how many times she's been to Panama. But when she came to Panama, she was blind in one eye. She couldn't speak, and she could only use one leg. And after a month of treatment, she was down for a month. After a month of treatment, she was walking, and she got her vision back, and she was able to speak for the first time. You can look her up online. She probably has hundreds of videos.
Are there any conditions in which stem cells don't work or you wouldn't recommend?
Well, there are a number of conditions we don't treat. If we do not treat Alzheimer's, we do not treat Parkinsonism. And I'll get into why here in a minute, but, I'll just stick with that for a second. So once you get diagnosed with Parkinson's disease, about 95 to 99% of the cells that produce dopamine in your brain are dead. So that means we're weaning along this little tiny population. And I think ultimately there needs to be a cell replacement therapy for that. But we want to treat people. We only have so much space. We only have so much time. We want to treat people that we know that at least we have a very strong suspicion that they're going to get much better and it'll have a persisting effect. With Parkinson's, my mother had Parkinson's, and we treated her like 5 times, and she got great results for 3 or 4 months. Of course, I have different resources levels than other people, including having my own clinic. But we don't want to be filling up chairs with people that have to come back every 3 or 4 months, and it's financially impossible for most people. But also, it's taking the chair of a kid with cerebral palsy or a kid with spinal cord injury that could be walking, and we don't get the level of benefit for the length of time that we would feel comfortable with actually charging these people to come down. So we just don't take those. There are clinical studies that are going on. I think there's one at University of Miami right now for Alzheimer's, and they're doing multiple treatments. And I haven't seen the results yet, but there are clinical trials in the U.S. There's also one in Parkinson's. So we just steer clear of those because we don't want to waste their time. We don't, hearing loss, I can speak from experience because COVID-related hearing loss. I lost all the hearing in my left ear, and nada. Like I've injected MSCs in there. I've injected magic juice. I've injected amnion. I've injected about everything you can that from a placenta into my ear. But anyway, deafness doesn't move the needle. We've had some people with neurologic deafness, but my deafness is from the hair cells in my cochlea, and I've had no movement of the needle. We don't make penises larger. We don't create new bladders. We don't create new organs. You know, the cells were in the early days. We used to think, oh, they're multipotent and they can do all of these things. They can become bone, muscle, cartilage, even liver. They can become pancreas. They can become all these different things. But in the human body, they don't work like that. In fact, Arnie Kaplan, he wrote an article. Ugo, were you on the article on changing the name? Oh, okay. Well, Dr. Barrera, he is a colleague of ours and Dr. Kaplan's, but he wrote an article, Mesenchymal stem cells, it's time to change the name. And he details how originally they can, and this is how he put it, we can make them dance in the laboratory, but then when we put them in a mammal, they don't become anything. But they're effective, they're useful through their secretions, and it has nothing to do with cell replacement. So we get some crazy applications for different body parts and stuff like that. We're not in the body part world. We're in the anti-inflammatory world. We're in the stimulating regeneration world, and we're in the immune modulation world. That's predominantly what we treat.
On that topic, how do MSCs work in our bodies? We literally got a question here. How do they know where to go?
Well, they have these receptors on their surface that look for inflammation. You know how Velcro works? There's a hook and there's a loop. One side's the loop, the other side are hooks. Just think of the MSC as having hooks on it, and then when a cell's in trouble, when a tissue's in trouble, it starts popping these molecules on the surface that say, hey, I'm in trouble, and the MSCs just land there, and then they respond to the environment, and the more inflamed the environment is, the more they respond.
Among all the things that you've been treating, you've mentioned several. What is one that you're most encouraged about?
Well, I think, DMD, Duchenne muscular dystrophy, we actually filed an IND with the FDA to do a clinical trial. We have a principal investigator she's done 35 FDA trials, Dr. Deanna Castro. So we're waiting on approval to go ahead with that study, and we'll see what happens. But I think we have a pretty good chance because we have treated several DMD patients, and we actually had compassionate use trials in the past. So I think we have a pretty good shot with FDA on that. So I would say like the autoimmune conditions are the most autoimmune and heart failure to me are the most gratifying because with heart failure they're not going to get any better. Mm-hmm. And a lot of the autoimmune conditions are not getting any better. We're looking at whether we should do rheumatoid arthritis or Hashimoto's, but we're working very closely with this rheumatology group in Dallas area, and they've done hundreds of clinical trials. Like they were one of the clinical trial sites for Humira and Enbrel and all those, those DMARDs, the drugs for modifying arthritic symptoms. And what I was astounded to find out is how unsatisfactory the standard of care is for rheumatoid arthritis. I thought you have the biggest drugs that we have are those DMARDs, the antibodies that are produced. And by talking to this expert, I got a big lesson on how ineffective they are.
Wow.
And so those stand out to me as far as the things that are most gratifying to treat because almost all of them get better, and it's just so simple, It's elegant, and it's like, God gave us these cells, and all we're doing is we're shepherding them from one place, growing them up in numbers that could treat more people, and then shepherding them into the body. That's all we do.
You've mentioned cerebral palsy a few minutes ago.
Mm-hmm.
Do you have anything to share in that regard?
We've treated several cases, and the thing to share is the younger the better, but at the same time, we've had a lady in her 20s that was treated and had shockingly incredible benefits. So it's very difficult because cerebral palsy is just it's a basket diagnosis for somebody that had a hypoxic event at or near birth, and the amount of damage that can occur ranges from very little to being completely incapacitated, typically physically. Yeah.
Let's move a little bit into the patient experience individually. So how do you determine the number of cells that each patient should get?
Well, we've determined what we think would be best for the patient. We don't know if it's optimal without doing a lot more work. We don't know what is optimal. I can tell you that in the Miami study where they gave 200 million cells, there was no benefit. Where they gave 100 million cells, there was plenty of benefit. We actually settled on 120 million being adequate for the reversal of inflammation for 95% of the population. So we stick with that. With MS, we give 140 because typically they just need a little bit more help. They're more, particularly if they're brittle MS, meaning, with MS you get lesions in your spinal cord and your brain, or one or the other, and they come and go over time. And in the early stages, that's called relapsing remitting. And then when they don't go away, that's called secondary progressive. So if you get somebody with secondary progressive, Holly was secondary progressive 12 years after her diagnosis, she was secondary progressive. So some of those lesions are just brittle and they're not going away. Our body has this capacity to fix those demyelinated, basically those lesions, which the lesion's there because the immune system has attacked the myelin, that's the insulation on the nerves in the brain, and that's why there's a lesion there. And if the lesion has been there and hasn't resolved on its own, that means they're running out of capacity to put more insulation on, which is called remyelination. And so those are more difficult, so we usually give a higher dose for that. But we also know that we give multiple doses because the vast majority of people don't have symptoms if you break up the dose. And if you give too many at one shot, you'll wind up with symptoms, a lot more symptoms, a lot more side effects. So we've always kept the maximum dose to 40 million because when we went to 50, we started getting, you could see the uptick in the percentage of people that had side effects, and we want it to be as easy and as enjoyable as possible. So we limit it to 40 million at a time.
A really popular question is, how long do these cells last?
That's a great question that I don't have an answer for, but we've never dissected human beings, after we've given them the treatment. So it's impossible for us to know exactly how long they last. There are some animal studies showing that they hang around for 3, 4, 5 months. All I can speak about is the effects. I can tell you how long the effects last for different conditions. So for example, there was a study done in type 2 diabetics, and it was like gold standard randomized controlled trial, and they gave one group MSCs, and oddly, and this is after we've been using 120 million for 8 or 10 years, these guys did this study, and guess what they used? 120 million cells. I literally don't know how they came up with that number, but it might have been published in some of our papers beforehand, but they gave them 2 rounds of 60 million cells. And what happened was their blood sugar normalized, their hemoglobin A1C. These were all type 2 diabetics that were insulin dependent. And the vast majority of them went off insulin. The global insulin reduction was like 85%. So not everybody got off insulin, but the ones that were on insulin, they were able to take a lot lower dose. And then the benefit, it lasted the full 3 years, meaning it was better than the placebo arm for all 3 years. But at about 18 to 21 months is when you saw that was, it went down like 3 months, but then it starts leaking back at about 18 to 21 months. So I tell, if you're worried about insulin resistance or metabolic syndrome, all that sort of thing, and I have the worst family history of diabetes of anybody, my dad, my paternal grandfather, my uncle, my maternal grandmother, cousins, it's just terrible. Like you're going to get the diabetes, so. But my hemoglobin A1C is 4.8, which is really good. So diabetes, kind of like high sixes, you're kind of in pre-diabetic, and if you're in the high sixes, these people were at, the mean was 7.5, and they dropped to 6. So you get a 1.5 reduction in hemoglobin A1C, which is enormous for preventing, the nerve damage, the kidney damage, the heart damage that occurs with elevated blood sugars. And so people say, well, how often should I get treated? I'm like, well, I don't know. How old are you? What are your comorbidities? But I can tell you from a diabetes standpoint, from a metabolic health perspective, that 18 months, 21 months sounds pretty reasonable. Now, for genetic diseases like for Duchenne muscular dystrophy, when we did our first human in the U.S., which was the first use of umbilical cord cells in the U.S., culture expanded MSCs, what we use in Panama. It was the first treatment of a human in the U.S., and we treated a young man, and we had applied for 3 years, and we were treating him every 6 months. So when we first did the plan, we did every 6 months. And the most sensitive measurement for this guy was his breathing, because he was already 22 years old. So he'd already lost most of his muscle, and his breathing was compromised. And we followed every week his pulmonary function. And what happened was it went up after, within a few weeks, and then it started declining at about 4.5 months. So then after 2 rounds of that, we showed the FDA, hey, maybe we could do it every 4 months. So we switched every 4 months, and then instead of him declining, he was actually he basically had at a high level for basically the rest of the trial.
Wow.
So for a genetic disease, I would say every 3 or 4 months is my experience, or our experience.
Thank you.
Yeah.
I have 3 more questions for you, but before I get into that, I would love you to share Corey's story.
He probably weighed 120 pounds, skin and bones, and he had all kinds of gut issues, so he couldn't absorb nutrients through his gut because he had an autoimmune condition that basically inflamed his gut, which is very thin, and there's only 1 cell layer between the food and getting inside, but his was so inflamed he couldn't absorb any nutrients. And then there's a picture of him like a year later, and he looks like Mr. Atlas or something. But anyway, we do a lot of gut issues resolve pretty well with the cells too.
Amazing. I would love to talk about safety. Can you elaborate on that?
Sure. Well, just looking at our safety record, I would also like to give credit to Dr. Kaplan for his basically, I would never had the cojones to ever try these in a human if they hadn't already done it at Osiris in Ohio. So, but out of 50,000 infusions, we never had even 1 serious adverse event. So that's pretty safe in our hands. And the meta-analyses, actually, Dr. Barrera was one of the authors on the first paper you put up there with Dr. Kaplan on the meta-analysis of the safety. People worry about cancer a lot, and I used to be a cancer researcher, and that's how I actually got into this field. We were doing therapeutic cellular cancer vaccines for cancer patients. But I was worried about that too. Do they cause tumors? Well, there's no evidence that they cause tumors. In fact, there's a paper that the title of it is Adipose-Derived MSCs Promote and Umbilical Cord MSCs Inhibit Tumor Growth in these animal models. And then there was further a study at Kansas State University. I grew up in Kansas.
There's somebody else from Kansas.
Go Wildcats. Here. Anyway, there we go. We shouldn't admit that in public, right? It's really hard to get the girls when you tell them you're from Kansas. But, anyway, they did a study there, and they did breast cancer, mammary cancer in rats, and they took umbilical cord MSCs and they injected them either 1 into the tumor or 3 IVs in the tail vein, and but the ones at placebo, the tumors just grew and grew and grew until they had to sacrifice the animals because the tumors were so big that the ethics committee wouldn't let them keep them around. And then it didn't matter which way they gave them, the tumors were like here, and then they just disappeared. And then the other way, it was, they're pretty much the same. It didn't matter whether you injected in the tumor or gave them IV. And so their hypothesis was that some way or another, the umbilical MSCs induced the death or gave the right conditions for the cancer stem cells to be killed. Because you can kill all the cells, and you got one stinking cancer stem cell, and then comes back. And in this case, these animals were tumor-free for the rest of their existence. So I'm very interested in a cancer protocol, and we're actually talking to different universities and hospitals right now about doing a trial in patients who have cancer cachexia, which we've treated 3 patients with cachexia, and we weren't treating their cancer, but we were treating their cachexia, which is the weight loss, the wasting. I don't know if you're old enough to remember the AIDS crisis before we had any answers for HIV. There were these guys that looked like they were going to die, and they did. But that's the muscle wasting that occurs, and it happens in cancer patients. It can happen in really severe advanced rheumatoid arthritis cases, but the global incidence is pretty high of cachexia. So I'm not afraid of that. I mean, I have no fear of that. And in fact, we're looking for an epidemiological partner to go and look at all our patient data because our cancer numbers are very low, given if you take patient years and you look at the number of cancer patients that have come out of that have been treated, it's very, very low. And I think we may be helping with the body's ability to repair itself. I wrote a paper years ago. It was basically cancer is a non-healing wound or something along those lines. And I gave examples of all the different cancers. The lung cancer, usually the chronic irritation, stomach cancer, chronic irritation from H. pylori, blah, 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 all the examples of different kind of cancers being secondary to inflammation that you can't break the cycle of the inflammation. So ultimately what happens is the bone marrow spits out cells and then you wind up getting cancer because the local environment, the MSC population has been depleted. The body says, okay, we're going to die. Let's release the hounds, right? And so let's pull out all the stops. And to me, that's a fascinating area of study, and I look forward to doing it. I think we have time for like one more because I see people are just leaving in droves.
Well, we're in Miami, so...
and it's a beautiful day outside, so I appreciate
is.
everybody for coming and staying.
So talking about Miami, what is the current scenario of stem cell treatments in the United States?
Well, unfortunately, there's still none approved in the U.S. They're approved in Japan, in South Korea, in Canada, New Zealand. There are products that have been approved. None have been approved in the U.S. There's one product that's in Europe that just got across about a year and a half ago, but it's for a very small indication. I don't know what's going on in the U.S. I asked Arnie because in August, Mesoblast, which is, they own the technology that bought Arnie's, they bought that company called Osiris that Arnie started way back in the day, and they're using those cells, the bone marrow cells that he developed. They're using them, that's the ones that got the approval, and they're using them in Japan. But anyway, they bought that, Mesoblast bought that, the intellectual property, they bought the cell lines, all the data for FDA, and they had very compelling results in a phase 3 clinical trial for the treatment of acute graft versus host disease in children, which is a deadly condition. And I think about 73% of them survive if they got the cells versus none, And so, because they're at a place where they're in danger of dying. And 9 out of 10, they have the committee, 9 out of 10 of the committee members, and they're all doctors and scientists that are experts in the field, voted for the FDA. It's a non-binding vote, but 9 out of 10 said, yeah, approve it, and they didn't approve it. So I said to Arnie, I said, what's going on here, bro? It's the second to last conversation with him. We talked monthly, and he got sick basically at the end of October. He said, the problem is that there's so many indications that the cells are useful for. I think they're concerned about them getting an approval, and then they're going to be used off-label for all kinds of stuff, and they won't have control over it. That was his take, and I don't know, but that was the Godfather's take. So,
tell us about your facility and what's next for you. Are you expanding? Are you focused in Panama right now?
Yeah, well, we're looking to expand to a couple different places, and I can't tell you where because we're in the middle of negotiations, and we have protocols that are under review, and I don't want to want to upset the apple cart in any way, shape, or form. But we do plan on expanding. We have, the company in Dallas. We manufacture cells there. The company in Dallas, we got a grant for doing this DMD trial. We're looking for a grant for doing the next autoimmune condition, and our hope is that the results will be compelling enough that we could get an approval someday. And so there you go.
Fingers crossed.
Fingers crossed. That's why I'm alive, apparently.
Yeah. Well, talking about life, you've impacted so many lives, and you've met thousands of people. How does that feel to be here right now at this
Well, it's the
stage?
best. Like I said, when I sat down and when I got up here, it's just so awesome to see so many people getting better, and I have so many. I'm blessed with so many friends from all the years that we've been doing this, and it warms my heart, and it's what keeps me going is people getting better and making friends along the way.
Awesome. We're going to wrap up here so we can actually interact with each other.
Okay, cool. Thank
Everybody,
you guys
please,
so much
a round
for
of
coming.
applause for
Yeah.
Riordan.
Thank you.